Flowering in plants is affected by a change in photoperiod. There are two different kinds of plants affected by this: short-day plants and long-day plants. Short-day plants require light periods shorter than a critical length to flower. They mainly flower in late summer, fall, or winter. Long-day plants require a light period that is longer than a critical length to flower. They mainly flower in late spring or early summer. Phytochrome entrains circadian rhythm in plants. This mechanism involves turning cellular responses on and off by means of the PR to PFR switch. In darkness, the phytochrome ratio shifts gradually in favor of the PR form, partly as a result of turnover in the overall phytochrome pool. The pigment is synthesized in the PR form, and enzymes destroy more PFR than PR. In some plant species, PFR present at slowdown slowly converts to PR, but upon illumination, the PFR level suddenly increases again as PR is rapidly converted. This increase in PFR each day at dawn resets the biological clock. The environmental stimulus that plants use most often to detect the time of year is the photoperiod, the relative lengths of day and night. Leaves detect change in photoperiod and produce signaling molecules that cue buds to develop as flowers. The hypothetical signaling molecule for flowering is called florigen. It is unidentified. Photoperiod and internal signaling molecules are necessary for flowering. The outcome is a transition of a bud's meristem from a vegetative state to a flowering state. Water balance in plants is regulated by stomata. About 95% of the water a plant loses escapes through stomata, although these pores account for only 1 to 2% of the external leaf surface. Each stoma is flanked by a pair of guard cells. Guard cells control the diameter of the stoma by changing shape, thereby widening or narrowing the gap between the guard cell pair. Under the same environmental conditions, the amount of water lost by a leaf depends largely on the number of stomata and the average size of their pores. When guard cells take in water from neighboring cells by osmosis, they become more turgid. The guard cells bow outward when turgid. This bowing outward increases the size of the pore between guard cells. When the cells lose water and become flaccid, they become less bowed and the pore closes. The opening of the stomata result in water loss from evaporation, and the closing of stomata help keep water in. The sensors that control thermoregulation are concentrated in the hypothalamus, a region of the brain near the ears. The hypothalamus contains a group of nerve cells that functions as a thermostat, responding to body temperatures outside of normal range by activating mechanisms that promote heat loss or gain. Warm receptors signal the hypothalamic thermostat when temperatures increase. Cold receptors signal when temperature decreases. At body temperatures below the normal range, the thermostat inhibits heat loss mechanisms and activates heat-saving ones, such as the constriction of certain blood vessels and the raising of fur, while stimulating heat-generating mechanisms such as shivering and non-shivering thermogenesis. In response to elevated body temperature, the thermostat shuts down heat retention mechanisms and promotes the body cooling by vasodilation, sweating, or panting. In the course of certain bacterial and viral infections, mammals and birds develop fever and elevated body temperature. A variety of experts have shown that a fever reflects an increase in the set point for the biological thermostat.